Dr. McCurdy. Christopher McCurdy is going to talk to us about Kratom, if I'm saying that right, or Kratom, Mitragyna speciosa as a potential therapy for opioid dependence. Dr. McCurdy is a medicinal chemist, a behavioral pharmacologist, and pharmacist whose research has focused on the design, synthesis, and development of drugs to treat pain and drug abuse. He has studied for over 20 years. His research is focused on opioid, neuropeptide FF, and sigma receptor ligand probe design, synthesis, pharmacological evaluations, and development. He is an internationally recognized expert on Kratom, and that is under investigation for opioid withdrawal syndrome. A significant portion of his career has been dedicated to generate and optimize selective ligands, which could serve as critical experimental tools, and more recently, as potential medication development leads to attenuate the effects of cocaine, methamphetamine, and pain. He is also currently president-elect of the American Association of Pharmaceutical Scientists, and also serves as the co-chair of the Special Interest Group on Drug Design and Discovery of the International Pharmaceutical Federation, and a lot more. He has a very long resume. That's just a fraction of it. So welcome, Dr. McCurdy.
Well, thank you, Dennis. Thank you, Annette. Thank you, everyone, for making this possible. This is really an amazing invitation for me as well. I'm not a person that's out amongst the healers and in the jungle and really finding these things. I was a traditionally trained pharmacist who got really interested in natural products, went to graduate school, worked with Lobella inflata lobeline, a natural product from Indian tobacco, for my dissertation work for treatment for nicotine addiction and Alzheimer's disease. And then went on to do a postdoctoral fellowship at the University of Minnesota with Philip Portuguese, who's been one of the real fathers of defining opioid receptors through antagonist probes for each of those subtype opioid receptors. And I just thought, I need a plant. I need a plant that I can find that I can work with. And so as I started my career at the University of Mississippi, I actually started working on Salvia divinorum about the same time that Brian Roth published a paper with Daniel Siebert on its activity as a kappa agonist. And so we actually, Brian Roth and I got funded at the same time to do that work. He did it in a different direction. We did it in a little different direction. My interest was how could a non-nitrogenous compound interact with an opioid receptor, because for years that just shattered the understanding of what was going on. And so we worked in that area for quite a while, did a lot of chemistry, did a lot of site-directed mutagenesis to understand the interaction with the protein. And lo and behold, I was invited by the National Institute on Drug Abuse to go and talk on natural products as analgesics. And in that research for that presentation, I came across Mitragyna speciosa. And Mitragyna speciosa more or less found me, I think, and I found it. And so I'm going to take you on a little bit of the journey that it's taken me on over the last, I'd say, 13 years. And ultimately has resulted in a very reverse of what most of you have talked about. I finally, for the first time about 6 weeks ago, made it to Malaysia and got to experience the actual plants. So let's see if this works.
Trouble here. It's not outputting to the screen, I guess.
Okay. Anyway, I hope I can show you a little bit like Nigel did very brilliantly on how he developed the product. We've been working very much along the same lines, but nowhere near as far advanced. And let's see if we can rock and roll. Okay, so Mitragyna speciosa is in the coffee family, in the Rubiaceae family, and it's named Mitragyna basically from the shapes of the leaf. So the shape of the leaf resembles that of a mitre hat of a bishop, and so this is where the Mitragyna genus name came from. There's different types of leaves. You can see a green vein, a red vein, and a white vein that are popular amongst the users. But this is a picture of the plain green vein here. It's found primarily in Southeast Asia, where it's indigenous, growing in Malaysia, Thailand, and Indonesia. It's also found in various areas around there. And different species of Mitragyna are actually shown in India and Africa as well. So mainly along that tropical plain. And the leaves have traditionally been used for opiate-like effects, but also for stimulant-like effects. And it is referred to as kratom in Thailand and biak-biak or Kratom in Malaysia. And so far to date, there's been a little bit over 40 alkaloids isolated out of Mitragyna speciosa itself. And just to give you an idea on the map of where we're talking about, most of the work and the area that I'll talk about here is in the upper part of Malaysia, in this area, and the southern part of Thailand. And so that's where most of the plants and materials that we've been dealing with most recently. Interestingly enough, all the materials that were available in the United States and probably still available in the United States today are most likely coming out of Indonesia. So it could be slightly different products than what we're dealing with. This is a picture of the tree in the flowering mode. It's actually right at the end of the flowering season right now. And this is a very one time a year event where you get to see the flower. And then the flower drops its petals and turns to a ball fruit. And I don't know if I have a larger picture of that, but these trees can grow all the way up to 15 meters in height, so they can become very, very large. In speaking to the native healers and the researchers in Malaysia, it takes about 3 years for a tree to really start to produce the profile of alkaloids that would be a benefit in a user. So it was interesting to us because I'll talk to you a little bit about cultivar that we tried to start and what limited success we had with that. So this is another picture. Many times these trees are growing in wet areas, so it's not unusual to see them near rivers or creeks, but this is just another site. So the earliest report that we could find in the literature, in the Western literature, is actually from the 1800s, and so this is not having the history that a lot of the products that we've talked about, at least a recorded history, and in the literature. But it was described first as an opium substitute, and the opium substitute was really utilized as a opiate suppressor, opiate withdrawal suppressor. And so this is very similar to like methadone or buprenorphine that we use in the United States for opioid withdrawal today. It was really stated that these effects were achieved through chewing the leaves. So leaves would be pared into quids and then chewed. This would give a stimulant-like effect, actually. Or it's drank as a tea. So the fresh leaves would be brewed into a tea. And what we've found to be interesting is that in Thailand, the traditional use is through chewing the leaves. And just over the border in Malaysia, the traditional use is by brewing it into a tea. So very different method of use, but very similar outcome. It seems to have this more stimulant-like effect, though, when it's chewed. And a lot of the laborers use this to be able to tolerate the sun and the heat and work longer hours in the fields. In the real traditional use of it in Malaysia is really the leaves are picked fresh in the morning. Those are then brewed for about 2 or 3 hours in boiling water. So a decoction is made, and then those are concentrated down even further, and then sometimes put in and stored in the freezer until they're needed for use. And when they use it, they then just dilute a half a glass of the juice with a half a glass of either warm or boiling water again. These are pictures of some of the fresh juice of how they actually store them today. So they'll brew them, put them into bags, and just toss them in the freezer. The benefits of modern living, right? So to say where we got sort of interested in this was really from a paper that was published showing some Thai abusers and Thai addicts in 1975. And this was a study that was really the landmark and very first study to show some of the human use of kratom. This was 29 males and 1 female that were interviewed basically through a survey in Thailand. And the ages range from 31 to 77 years old. And what's interesting is most of these people have been using it for at least 10 years or more. And what their use level is usually 3 times a day, so morning, afternoon, evening, and they would use it either once at those sittings or 2 to 4 times at each sitting. So it just depended on their level of need or their level of interest, I guess. Physiological effects, again, just a few minutes after consuming this, they feel happy, strong, active. They possess a very calm mind, but they prefer to stay alone. Actually, many of the men reported to use this to prolong sexual intercourse. And that's been a consistent theme that's shown up in more modern surveys as well. And so there is some interest in that area, particularly as we move down the road and looking at developing in different therapeutic areas. But right now we're mostly focused on this opioid crisis that we're facing in the United States and seeing what we can do with this for that purpose. Physiological effects, though, long-term physiological effects from this, the addicts will become thin and the skin will darken on the face. And generally it's on the cheeks, and it varies again from person to person as to how dark the face becomes and how long the exposure has been. It does produce a dry mouth, constipation, which would be pretty typical what you would think of if you're involving opioid systems. Main 2 problems with prescription opiates are constipation and respiratory depression. And we'll get to the respiratory depression aspects of this as well. And one thing that really caps this as potentially having an abuse potential is that it does have a withdrawal syndrome associated with it as well. Okay? So does coffee. If some people don't have coffee in the morning, they get splitting headaches. The withdrawal syndrome of kratom is not too different from that of coffee. So the withdrawal is very mild. There is some aggressiveness. There is some agitation. In some people that are very heavy users, you can get muscle jerking and movements. But really, if you think about this compared to a traditional opioid withdrawal, this is really nothing on the order of concern. So a couple of cases in this study of addicts that were of interest were really a couple of cases of psychoses. So there was a 50-year-old male subject that had been using for 35 years. And he had reported that he was having convulsions and mental confusions, cloudy consciousness, and he had delusions and hallucinations. A 32-year-old Thai laborer using for 10 years had confusion and a little bit of ideations of persecution and concern, maybe a little paranoia type feeling. And then the third one was a 31-year-old who had used for 17 years that was just having also hallucinations, delusions, and confusions, dizziness, and headaches. So these were 3 of the extreme reports out of that 30-group study. So this was about 10% of the people in the study that had some sort of more severe side effect or reaction. And you can look at this. This is a pretty extensive usage history. So when you're talking about 10 to 30 years of use, there's no telling that if it's from the plant or if it's just the individual that ended up having some sort of psychosis normally. One other thing that's of note, very good interest, is that the use of this is really accepted amongst the male population in both Thailand and Malaysia because it was used for vigor, for improving that working ability in the fields. Because of that, it was very frowned upon for women to use Kratom. And so there are women users. There was one woman user in this study that was interviewed. But most of the women, even to this day, are more likely not to admit that they're using because they don't want to have a stigma associated with them. So it's very hard, even for the clinicians that are getting out into the communities where this is being used in Malaysia and Thailand right now, to interact with the females and get any information out of the female population. So hopefully that can change. One thing that's been long associated with the use of this is not just that stimulant-like effect, but also a pain-killing property. So this analgesic effect that has been associated with it for as long as it's been used as well. And really the literature lay pretty dormant with Mitragyna speciosa mentions until about the 60s and early 70s, when a pharmaceutical company, it's now GlaxoSmithKline, but it was SmithKline in French, I'm sorry, in the 70s. They had actually done some studies with pure mitragynin. So mitragynin is the major alkaloid, and I'll speak about that in a moment. And up to that point, this was the most comprehensive study that appeared in the literature on mitragynin, the purified major alkaloid from the plant. And it was thought that major alkaloid was the one that was responsible for all the activities because it was the major component. They found that the analgesic effect was comparable to that of codeine, so it wasn't really a powerful analgesic. But many of the people that use it traditionally say it's very good for warding off any types of pains, especially muscle pains, any types of labor pains that would be from working, overextension of the muscles and those types of things. This was really good for treating some of that pain. What was interesting in their paper, though, was that they found mitragynin could not be antagonized by nalorphine, which was at the time one of the opioid antagonists. So now we know that nalorphine is not a pure opioid antagonist, and so this confused the literature for quite a while. Interesting thing that's not up here from this paper was that they also did some maximal tolerated doses going up to almost 1 gram per kilogram of pure mitragynin with no side effects in the animals. Unbelievably safe compound, at least as far as they noted. They also noted that it did have slight respiratory depression at very high doses. They also noted that it had GI constipating effects at very high doses. But again, we're talking about very high doses, okay? So not near the therapeutic effects. And this will come back a little bit later on as we start moving through this a little more. So this is where I kind of found the literature when I was asked to make this presentation at the National Institute on Drug Abuse. And so then we jumped into the scene and said, hey, let's see what we can get. And so we bought premium Mitragyna speciosa from the internet, like any good scientist. You know, we went out and just found our fastest source, which later on came back to bite us because we had no control of the biomass that we were getting. And so I'll bring that up a little later. But interestingly enough, what we were getting was very good material. It was authenticated. We fingerprinted it. We did all the work. All the analytical work. Well, all the analytical work was done by my beautiful wife in the back there. And she's done a lot of the analytical fingerprinting. I'll talk about a little bit as well. So the other thing that we did at this time, we were buying such large amounts of leaf material at a time when nobody had even really heard of Mitragyna speciosa in the United States, that it flagged the guy at the company to put me in contact with the supplier in Indonesia. And so we actually immediately got into contact with the supplier in Indonesia. He was interested in why we were buying so much, what's going on. And so I was explaining to him that I was really interested in the plant from a scientific perspective, and we were going to try and do lots of extractions and isolate compounds and study the compounds' pharmacologies and find out if there was anything really to these claims about opioid withdrawal treatment or potential new avenues for analgesics. And so he was excited from the standpoint that he said, well, okay, I'll send you a couple of trees. Great, why not? So he sent us a couple of trees. And the beautiful thing at the University of Mississippi is they have the National Center for Natural Products Research in the United States, and they have an arm of the USDA's Agricultural Research Unit embedded in that center. And so with that, we have botanists, we have all kinds of excellent trained plant scientists that we were able to take those 2 trees and propagate those to close to 100 trees in very short order. And we grew those trees up very nicely, actually in a greenhouse right behind the chancellor's home. He had no idea. There was also 200 Salvia divinorum plants growing in that greenhouse behind his home, with no idea of what was back there. So we actually got to the point where we outgrew this small greenhouse on campus, and then we had a field station, a very large field station out in the country, where we moved everything that we had. We had to move all those trees inside during the winter because the trees will not survive freeze at all, not even for a short period of time. We did get brave after about 2.5 years and about a 4-inch, 5-inch diameter trunk. We planted one outside and it didn't survive the winter. And the winter in Mississippi is very mild. You may get one snow. And if it does snow, it's gone by lunchtime the next morning. So does not survive the cold temperatures at all. So what was interesting to us, we started getting enough mass that we could harvest our leaves and start looking at everything. We had the plant at least authenticated down to the cellular level that this was a Mitragyna species. We didn't know if it was precisely Mitragyna speciosa because we started to raise doubt when we couldn't find very much of the active compounds in our matrix. This came into play when I found out that the trees need to be probably 3 years old before they start producing the proper profile. And so we were looking at this just 2 years after we had started this project. It was a little bit frustrating. Also, the scientist at the USDA that was helping us with this actually left and returned to Brazil. And so we didn't have anyone to really take care of our plants anymore. So we just decided to do a wholesale sort of extraction. We looked at roots, we looked at bark, we looked at stems, we looked at leaves, we looked at everything. We found very little to no Mitragyna anywhere in the extract. So it was a little disappointing, and as a young, naive scientist, I kind of was just disappointed and said, well, let's just chalk it up to we didn't get the right stuff. Lo and behold, come to think about it a couple years later after we got rid of everything, that would have been a perfect placebo to study. But, oh well. So what we did was we wanted to look at the extracts because remember, the pure Mitragyna itself was what was looked at in Mako studies at SmithKline and French. So what we did was we took extracts from the premium Thai plant material that we bought, and this is 500 milligram per kilogram organic extract that we used with essentially just methanol. And then the Mississippi extract, which I have labeled ME, and this is a hot plate latency, so this is putting an animal on literally on a hot plate and waiting for them to start reacting to the heat by lifting their paws or start licking their paws or start moving. You can think of it as going on asphalt in bare feet, and you don't really notice it in the middle of the summer that the asphalt's hot at first, but by the time you get to the middle of the road, you're trying to quickly get to the other side. So that's a measurement really of supraspinal analgesia, so that you're recognizing that there's pain. It's not a reflex action. We also took this through a tetrad assay because we were looking at effects of cannabis substitutes at the same time. So the University of Mississippi also has the federally funded, the only federally funded marijuana farm in the United States. It doesn't produce the greatest quality marijuana compared to some of the competitors that have come up in California and Colorado and other places. But it's still a mainstay of the work that goes on there. And we finally got a grant to look at natural products for neuroscience while I was at the university. And the Natural Products Center at the University of Mississippi was really focused on antibacterials, antiparasitics, antifungals, and antivirals. And so a huge repository of plants, but never been screened for CNS activities. And so we had started to dive into a lot of the repository and screening those at opioid and cannabinoid receptors, as well as looking at plants on the internet that were stated as being cannabis substitutes or opium substitutes, of which Mitragyna was one. So we basically ran all of our extracts through the same paradigm. We started to look at this tetrad assay. So the tetrad assay is a standard assay that was developed by Billy Martin, a cannabinoid researcher at the University of Kentucky many, many years ago, where he showed that cannabis would drop body temperature. You would also have changes in locomotor activity, so the animals would decrease their locomotor activity. We would look at analgesic activity, and then we'd look at catalepsy, and catalepsy would be a sign of psychoactivity. And so what we're seeing here is just changes in rectal temperature at the very top with the premium extract and the Mississippi extract. Down here, locomotor activity seemed to be impaired at the higher doses with both extracts. So interesting that we were able to see maybe it's not an effect associated with the mitragynins, but maybe the change in body temperature was, because the difference between these two extracts was really that major alkaloid missing. So we went on to isolate several compounds that are already known in the literature. Mitragynin is here on the top left, and its famous partner, oxidative metabolite 7-hydroxymitragynin, which really has garnered a lot of the attention, specifically of the DEA of recent. And then coinanthine, so these are all coinanthine type alkaloids. Pananthine, asmalacine, mitraphyllin, and isomitraphyllin were compounds that we were easily able to isolate and identify out of the premium extracts. So we wanted to look and see if these had activities at opioid receptors, and so we screened these for affinities. We're not seeing the impressive affinities that Nigel saw at some of the targets, but mitragynin had about 600 nanomolar affinity for mu-opioid receptors, and mu-opioid receptors are the main protein that morphine would interact with. Delta opioid receptors are thought not to play much of a role in the actions of kratom or the individual alkaloids, and you can see it's at micromolar affinities, mitragynine for delta. And then kappa opioid receptors, which we talked a little bit about yesterday with Salvador-Ney, are thought to be involved a little bit in the analgesic effects as well. And so interesting to us was mitragynine was pretty weak. If you compare this to morphine, morphine is about 60 nanomolar at mu opioid receptors. If you want to talk about fentanyl, we're talking about subnanomolar affinities, so very much more potent compounds. But what was interesting to us was 7-hydroxymitragynine, a very minor component in the plant, so it makes up less than 1% of the total alkaloid extract. This one had 100 nanomolar affinity for opioid receptors, mu opioid receptors, and much higher affinity at delta and kappa opioid receptors. And so we thought this was fairly interesting as well. And then in some previous SAR work that had been done and reported in the literature, corianthine, which lacks the O-methoxy on the indole ring, actually was shown to be an opioid antagonist. And so we don't know, we didn't really pursue that any further, but it had better affinity than mitragynine at the mu opioid receptor as well. So the other compounds were not very interesting as far as opioid receptor ligands go. We did want to make sure that we had the right structure, so we got an X-ray crystal structure of the material that we isolated. It was the correct material, so we were happy to keep moving on. And then we wanted to look at these in analgesic assays again, just to reconfirm what Mako has seen in 72. Morphine is here on the left. Again, this is the hot plate test. And then we did various doses of mitragynine on the right, and you can see it's much less potent, much less efficacious as that compared to morphine. And if we combine those into one graph where you can directly compare the efficacies, you can see that morphine is much superior. However, we did go ahead and take naloxone, which is a pure opioid antagonist, to reverse the effects. And what you can see on the far right side there is where we were able to completely abolish the analgesic effect of mitragynine with pure opioid antagonist. So it is an opioid-mediated analgesic measure. There's another assay that can be done, and that's a tail flick assay, and this is a response to pain that's a reflexive response. So this is where you put an animal's tail in a groove and you shine an intense light beam on it, and after about a second that animal will literally flick its tail out of the way. If it's under the influence of an analgesic, it will stay there until the cutoff time so we don't burn skin or do any harm to the animal. But they will stay there for up to 15 seconds. And so you can see morphine performs extremely well in this. On the right, mitragynine performs not very well at all. And then when we look down even at the premium extract, the extract is containing all the alkaloids. So we do see a little bit better activity with the extract than we do the pure mitragynine. So this was another indication that there might be something special with the extract. So we came back to this 7-hydroxy mitragynine. There was a paper that came about the same time in the literature from a Japanese group led by Matsumoto that was looking at 7-hydroxy mitragynine specifically and its analgesic activity. And they reported intense analgesic activity. And so what's really amazing to me is the triangles are the 7-hydroxy mitragynine. The circles, the black circles, are morphine. So you can see 7-hydroxy mitragynine in these assays was much more potent than morphine. Okay, this was again, this was subcutaneous administration. And then what really floored me, and really as a scientist said, there's no way, this is oral administration. So if you look at how fast this molecule is working, within the first 10 minutes, it's at 100% maximal efficacy, orally. So we go back to subcutaneous, if we go back to subcutaneous, within the first 10 minutes at 100% efficacy. So morphine takes a long time before it starts to reach any type of analgesic efficacy when it's used orally. And part of that is because it goes through first-pass metabolism and intrahepatic recirculation, and the bioavailability of morphine and morphine analogs is miserable. And this is where having a pharmacokineticist and really studying a lot of the transport helped us tremendously. So we started to question this. We wanted to replicate it in our lab, and we did. I mean, I still couldn't believe it when my students brought me. We looked at this subcutaneously, orally, intraperitoneally, and IV, and the curves were all superimposable at every dose. I couldn't believe it. I've never seen that with any compound that I've come across. So almost identical to those that were reported previously. And what I want to show you is, and come back to this IV and oral pharmacokinetics of what was so important here. And so pharmacokinetics is just a study of what the body does to the drug. So we're all focused on really the pharmacology, the pharmacodynamics of what is the drug doing to the body. But we're interested from our standpoint of what's the body doing to the drug as well. And so this is Mitragyna, the major component of the plant. And this is what a typical pharmacokinetic blood level would look like. You take the compound orally, so there's nothing in the blood at time 0, and slowly over time, and this is 5 hours, okay, up to 5 hours, you start to get a peak at about an hour and a half into the blood supply, and slowly it will start to be metabolized and eliminated out of the system. Okay, intravenously, you're going to have your maximal concentration immediately after you introduce that drug into the vein. And so you can see the concentration goes from high to low. So the reason that I want to show you this is to show you that same graphs of the 7-hydroxymitragynine. And so we were so shocked by how fast this worked. This is oral and IV pharmacokinetics data of 7-hydroxymitragynine. I've never, ever seen another drug that shows this type of data. And what you can also see is the bioavailability of this compound, which I don't have the calculation here, but we're looking at concentrations in nanograms per mil. We're almost coming equivalent to what was injected straight into the vein, right? If we go back to this slide, this scale is not even reaching a little bit over 400 nanograms per mil, when that scale is topping out at about 5,500. Okay, so huge difference in pharmacokinetics. Now, I want to bring back the point that Nigel made earlier. These are the pure compounds. These are not in the plant matrix. Once these are all in the plant matrix, the story changes completely. And that's the beauty, I think, of these extracts. So certainly we have an orally active analgesic. This has gotten a lot more attention recently. This is, I believe, really what's driven the DEA and a lot of their concerns about banning this substance because of how rapidly the 7-hydroxy can be absorbed and effective. And I'll come back and talk about that a little bit later as well. But really the way we look at this is, one, this is a fantastic lead compound for development of new analgesics because you don't see the side effect profile that you see with morphine and morphine-type derivatives. And what we're starting to really learn is these compounds don't seem to produce respiratory depression like your typical opioids. So the main problem with death related to opioid overdose is respiratory depression. People stop breathing. If they're not treated with naloxone quickly, they will die from suffocation, essentially. One of the hot theories right now in pharmacology and opiate pharmacology is beta-arrestin recruitment. So this is a biased signaling mechanism. At the cellular level, this is a protein that is recruited by some compounds and not others. So interestingly, a lot of the traditional opioids do not recruit beta-arrestin. They signal through the traditional G protein coupled receptor pathway. It seems that the mitragynins recruit beta-arrestin, and some people are equilibrating that to the fact that they don't cause a respiratory depression. I'm not 100% sold that that's the the only reason. I think it has a lot to do with the way these compounds are interacting and their kinetics at the cellular receptor level in terms of their on-off rates with what's going on. So what about, we saw that this might have a better safety profile from that standpoint. But what about abuse liability? This was reported again by Matsumoto's group in 2008, where they took 7-hydroxymitragynine and put it into a condition place preference assay. So condition place preference assay is where you have a box with two distinct chambers, and there's a vestibule in the middle. So an animal, a rat, or a mouse can be placed in that vestibule in the middle. You open up access to the entire box. That animal freely roams around. Rodents are very curious. They explore everything, and you want to make sure that they basically have no preference or aversion to one side or the other. So the difference from one side to the other may be that you have vertical black and white stripes on the walls, and the other side may have horizontal black and white stripes. But there's a definite visual and sometimes tactile cue. The floor may be gritty on one side and smooth on the other. But there's a definite difference to the animal to those sides. Then after the animal has been exposed to that, you take the animal, replace it back into one side, and you couple that with the drug that you're interested in studying. And so you only confine that animal to one side. That animal, whether they feel good or whether they feel bad, they're going to remember pairing that with the cues, either the visual or tactile cues, so that when you take several days away, you put the animal back in, open up both sides. That animal is going to spend more time where it felt good and hopes that it's going to feel good there again. Or if it had a bad experience, like we've seen with some of the kappa agonists, it will be aversive and it will spend its time in the side where it didn't receive drug, but it didn't feel bad. So this is a condition place preference, condition place aversion assay. It works pretty well for drug-seeking behaviors. Doesn't really give us an indication of addiction, though. So what you can see here is as the dose increases from 0.5 milligram to 1 to 2 milligram per kilogram of 7-hydroxy, you get a significant effect greater than that of morphine or similar to that of morphine in a place preference assay. So these compounds, the 7-hydroxymitragynine definitely induces a place preference in rodents. So they will seek out, try to recapture that feeling again. And if we look at the 2 milligram, and I reviewed this paper, I argued with the 1 milligram dose that it had to be statistically significant because the bars overlap with morphine, but it got published anyway. If we go back to the doses, if you look at 2 milligram per kilogram dose, in both assays, it's active, but it's weakly active. So it seems to be having a euphoric or a drug-seeking behavior long before it's getting to the really efficacious analgesic doses. So this was a concern for us as we move forward trying to come up with some answers. Most recently, I started working with a colleague, Scott Hemby, at High Point University in North Carolina. Scott is an expert in speedball self-administration, so cocaine and heroin self-administration in animals. And so I started asking him, well, could we see if mitragynine or 7-hydroxymitragynine substitutes in your animals that are addicted to morphine or heroin and just see what happens? This is a key study for us in terms of the abuse liabilities. And this is the left side. So mitragynine, black bar here is saline. What you can see is as we escalated doses, and these are direct infusions into the brain, you're not seeing any substitution of mitragynine for morphine. And if we move over to the right panel, this is 7-hydroxymitragynine, and as the dose increases, the animals are working substituted readily for morphine in animals that were already habituated to morphine. And the reason it goes down, this is a typical inverted U-shaped curve, because once they get to the higher dose, they're fine. I don't need to take any more. Just hang out and feel good. So we wanted to look at, this was substitution. We wanted to look at it from a de novo, naive animal experience. And the more dose that we gave these animals access to, the harder they worked for getting in infusions of the drug. And so This definitely showed us that there was abuse potential with the 7-hydroxy mitragyna. So, okay, where does this leave us when we talk about opium and morphine? And now we've got conflicting data in a plant that's got compounds that are analgesic, maybe to the level of codeine. It's got a very minor component, less than 1% of the makeup, that has incredibly potent analgesic activity and abuse liability. But what happens when you mix it all together? So that's the question that we've been looking at and trying to address. And of course, I'm not going to go through any of this other than to state the fact that as of the latest data that I have in 2004, the United States consumed 99% of the world's supply of hydrocodone, and yet the United States makes up 4.5% of the world's population. And so you can see that even in the early 2000s, the opioid crisis was really starting to escalate in the United States, and this is where we jumped in to the path. Wanted to look at treatment programs to try to figure out what was already out there, what were our options to compete with. You have rapid detox programs where people can go in and be injected with opioid antagonists like naloxone. And within three days or so, they're pretty much back out to the street and detoxed. They say it's like going to hell and back, though. They said this is one of the worst withdrawal treatments you could go through. Most of the ones that are used and accepted, though, are methadone or buprenorphine treatments that can be prescribed as tablets. A lot of times clonidine is used, which is an alpha-2 agonist. This is to block a lot of the other side effects associated. And then, of course, that needs to be coupled with psychotherapy as well. So all of these treatments that currently exist have limitations, and they all have very high relapse rates. And so a lot of times the individuals will run into a crisis and they'll just stop taking their methadone, stop taking their buprenorphine, and go back to using again. And so we wanted to try to find something alternative to this. Methadone and buprenorphine are also full mu-opioid agonists, but they have different kinetics, okay? So they actually go onto the receptor at a very slow rate. They don't cause the euphoria that you get with something like heroin that is more like a rocket ship that gets to the receptor very quickly and causes euphoria. I teach my pharmacy students that this is the equivalent of a Ferrari versus a old Yugo from the good old Yugoslavian. If everybody remembers the Yugo, most of them don't, and I realize I'm getting old. So, so then we have to figure out some other cheap car. But anyway, one other thing that really interested me from the standpoint when we tested mitragyna and saw that it had low affinity. Not what we expected to see at opioid receptors based on what we had seen in the literature. I sent it off to Caliper Life Sciences NovaScreen for their CNS panel of targets. And for $10,000, you can test 90 targets and get back a graph that looks like this. So this is my $10,000 slide. And it confirmed two of the things we knew already. One is that it bound to mu-opioid receptors and kappa-opioid receptors. But the other thing that really excited me was it bound to adenosine A2A receptors. Adenosine A2A is the target for caffeine. So this could explain potentially some of the stimulant activity if it is acting like caffeine did. Adrenergic alpha-2s. Oh, I was excited to see this because clonidine is one of the drugs that's prescribed for opioid withdrawal. And so if this is activating alpha-2 receptors, you're reducing the amount of adrenergic system activity and potentially reducing the amount of side effects that could be there. Dopamine D2s also involved in drug abuse, drug euphoria. And then serotonin 5-HT2C and 5-HT7 also targets known to be involved with drug abuse. And so this, to me, looked like Mother Nature's perfect drug abuse treatment compound, at least opiate abuse treatment compound, because it had already employed some of the individual pharmacological targets that we've looked at over many years of treating opiate addiction. So I called up a physician friend of mine, Ed Boyer, who was the lead on this paper, started talking to Ed and asked him if he'd ever heard of Mitragyna speciosa. And we were already talking about salvia divinorum, and he was wanting to do human trials with salvia, which we never got approved, by the way. But I asked him if he had heard of kratom, and he said, nope, never heard of it. What is it? So we started talking about it. And I said, start asking your patients that you treat. And he said, everyone I asked that knew about it had taken it. I said, we're on to something. So what we did was a quick survey of addicts in the chat room. This is not scientific at all. But all we wanted to do was get a starting point for where we should do animal work from. And so what we did was we asked addicts that were in this chat room. Again, there's only 14 votes. In some of these, there's only 7. But if you've used kratom for withdrawals, how many times a day do you use it? So was it once, twice, or 4 or 5 times a day? How much do you use? And then have you ever stopped kratom cold turkey or CT? Yes, I had terrible withdrawals, 0. Yes, and my withdrawals weren't so bad was the majority of the answers. And then no withdrawals at all was the other answers. And so this gave us a really good starting point to start working from. And so what we did was we made aqueous extracts. We made tea just like they do, excuse me, in Malaysia. And just actually, we took the recipes off the internet of how people were using it. We made the tea. We lyophilized that tea, freeze-dried it. We then quantified how much mitragyna was present in a gram of dried powder. Then we started looking at how much 7-hydroxymitragyna was present as well. And so the ratio of mitragyna to 7-hydroxymitragyna has been a key interest of mine. And in the natural plant, it's about 23 to 1. So it's about 23 parts mitragyna to 1 part 7-hydroxymitragyna. And so then what we did was we looked at habituating mice to morphine. So we would do escalating doses twice a day for 5 days in mice. We'd start with 20 milligrams per kilogram twice a day and get them up to 100 to 120 milligrams per kilogram twice a day after a 5-day period. And then we could precipitate withdrawal, do the rapid detox essentially with naloxone. And then we can measure all of these different aspects. And so naloxone by itself is pretty similar to saline. So there's no just endogenous tone of withdrawal. If you block your own opioid receptors, you block your endogenous opiate system, you're not going to go into massive withdrawal. However, if the mice were habituated to morphine and we gave them a precipitated withdrawal, now we have a whole battery of side effects or behaviors that we can measure to see what's going on. So first one is locomotor activity. Locomotor activity is extremely decreased. This is because the animal's feeling pretty bad, presumably. They do a lot of jumping. We call this popcorn jumping. They'll actually, all 4 limbs will fire basically at the same time, and they'll jump like this. Paw tremors, they'll go through pretty much sit back on their hind legs and their front paws will shake. They do wet dog shakes. And by the way, this is over a 30-minute observation period is what we're recording. So wet dog shakes, they'll actually shake from head to tail just like a dog getting out of a pond, but they're not wet. Teeth chattering, you can audibly hear them chattering their teeth through this. And then they lose tremendous amount of weight during that time frame compared to normal mice that are just on saline that are actually in the stage of growing, and so they should be gaining weight. So we saw that. With the kratom tea extract just by itself, we saw very little side effects other than the fact that we did see wet dog shakes after we precipitated with naloxone. So there's something interesting going on just with the pure extract. And then when we gave the extract to habituated mice, so 5 days on morphine, switched them over to 5 days on kratom twice a day, and then we precipitated withdrawal. What you can see is almost all those side effects have been wiped out compared to just morphine precipitated withdrawal. We still have that wet dog shake issue, and what we think that is really a serotonergic mechanism that's taking place and is probably related to a lot of the other alkaloids that are present in the plant extract. We looked at this up to 2 grams per kilogram of tea extract, so they were getting 4 grams per kilogram per day of the tea extract, and that's what dose I showed you on the previous one. We wanted to look at methadone at 23 milligrams and 100 milligrams per kilogram as comparisons to pure mitragyna at 23 milligrams per kilogram. Remember I said this is about 23 parts to 1 part in the extract. So we took 23 milligrams is what we were figuring the animals were getting in a 1 gram per kilogram dose. And what we saw was really no side effects with mitragyna, and pure mitragyna really looks fantastic to detox animals from opiate withdrawal. So where do we go with all this? We've been working on this. We've actually started to look at talking to companies. We ran into problems, as I mentioned earlier, with our biomass supply. So you can't go to the FDA and say, hey, we're getting all our stuff off the internet. Let's go do a human trial. No, they weren't gonna go for that. So the contacts we've made in Malaysia lately to get access to controlled farms is really fantastic and really helpful for where we're going forward with controlled biomass sources. We have eliminated the wet dog shake. We've created a what I would call a decaffeinated sort of version where we've eliminated some of the alkaloids that we thought were causing those wet dog shake side effects from the full extract. This extract still has mitragyna. It still has 7-hydroxymitragyna present. That little bit of 7-hydroxymitragyna might be enough to act like that full opioid agonist that you get with buprenorphine or methadone to just satiate the addict enough, and then the other compounds may be helping in an orchestrated symphony to really calm the rest of the system down and then be able to eventually get them off. But we've got a fraction now that we think works that's ready for prime time once we get there. So it brings me to the last part of the talk, which is why does the DEA all of a sudden get so excited about this? Well, I have 2 reasons. One goes back to adulteration. That's the main one that Nigel brought up earlier. But the second one is this is a matter of being in the wrong place at the wrong time. Okay? So Kratom actually ended up on gas station shelves next to this thing called K2, spice, synthetic cannabinoids, and this other thing called bath salts. Okay? And so in gas stations, and literally, I mean, on my way home from work, I could stop at the gas station to this day and pick up all the Kratom products I want right now in the United States, and we'll get to that in a minute too. But I think what happened was there was such a push from the synthetic cannabinoids and the bath salts to make these criminalized and put them into Schedule 1. And what was sitting on the shelf next to them? Mitragyna, speciosa, and Kratom extracts. And funny, not funny, typical government bureaucracy has put Mitragyna speciosa, natural extract product, into synthetic ban bills. And I've actually seen articles written in newspapers and on television that have stated that Mitragyna speciosa is a synthetic drug that's being banned. And so there's a huge problem with the public knowledge and awareness of this. And the states that it has become outlawed in, it was simply clubbed in with all of those synthetic cannabinoids and other materials. So that's one thing, being in the wrong place at the wrong time. The second thing is adulteration. So I mentioned to you that the 7-hydroxy is this pretty powerful, potentially addictive opiate substance. And what we can see, and I'm sorry this is a little bit small, but this was just published in Journal of Medical Toxicology. Again, analysis done by my wife, Bonnie Avery, in the back. But in the natural kratom leaf, we have about 24 micrograms per milligram of leaf material of mitragyna. 7-hydroxy is about 124 nanograms. So really you're getting that 20-to-1 sort of ratio. if we look at the products that are available, and by the way, this was a drive home from work one day. I stopped in and picked all these up at $9 a pop. And we took them home. The reason I was interested was because I was getting phone calls from people and emails from people saying that their child was in a detox program all of a sudden, and all they've been taking is these products. And I said, that just doesn't make any sense. So we wanted to analyze and see what was going on. Interestingly enough, most all the products are only containing the compounds that you would find in Mitragyna speciosa. But what has happened is, in some of them, the levels of 7-hydroxymitragyna have been increased by several fold, 5 to 6 times what was existing in the normal plant material, and the amount of mitragynin has gone down. And so now we're starting to look at self-administration studies of mitragynin and 7-hydroxymitragyna together in that ratio to say, okay, where is the break point? As we start to decrease mitragynin and increase 7-hydroxymitragyna, where does that point come where the 7-hydroxy takes over and becomes a problem? So that's one thing we're looking at right now. That's just one of the adulterations. Okay, so adulteration with the natural product itself. The second thing is we've been contacted by emergency rooms around the country, and I'll try to go through this real quickly. This was a newborn that was born full opioid withdrawal. The physician said on a 1 to 10 scale, the baby was 11. Very sad. The mother swore she had never taken an opiate, but she finally admitted to being on Kratom. So I immediately asked them to send us a sample of what she was taking. We took that down to the analytical lab. Bonnie analyzed this. We got not only the leaf material that she was ingesting, but we also got the meconium sample from the baby to look at. So the first stool sample from the baby. Went through and did analysis. So this is just to show you that we actually really do analytical chemistry. These are mass specs of the standard compounds. And what we went through was looking at these standards, so morphine, hydrocodone, mitragynin, 7-hydroxymitragynin, 7-hydroxymitragynin. I guess we didn't do a good job purifying that, but it's mostly 7-hydroxymitragynin. And so we went through the material that she was taking. It came up negative for morphine, positive for hydrocodone. So the leaf material that she had was adulterated with hydrocodone. And there was no question. I mean, this is pure forensic analysis. Of course, mitragynin and 7-hydroxymitragynin were present as well. We wanted to look at the meconium sample. Meconium sample was positive for mitragynin and 7-hydroxymitragynin. Did not show hydrocodone in the meconium sample. However, we think that's due to the fact that this is going to be all eliminated through renal and kidney into the urine. So you wouldn't expect to see the pure compound or metabolite in the meconium sample. So that baby was treated based on the information that we fed back that they definitely had opiate exposure. That baby was treated and released and was fine. Eventually, we don't know down the road what's going to happen. Then the second case study was one that was admitted to the University of Colorado Hospital with seizures. She was in kidney failure, complete shutdown, denied consumption of morphine, but said she'd been taking Kratom that was purchased online. And she lit up the tox panel like a Christmas tree for opiates. And so we did the same thing. We asked for the samples. We verified that hydrocodone was present in the sample that she ingested. It was absent in our standard down below here. And then we could detect the presence of morphine as well in this sample, but it was below our limits of quantification, so we couldn't say how much was there, but there was definitely positive, qualitatively for morphine in the sample as well. So those are just a couple of things I think that have raised the bar and concern in the United States. We don't have a good mechanism to control these products that are being sold and being used, and what we've seen with what's available is really adulterated and really abusing the plant more than the plant being abused. And so I really want to go back and get some standard regulations on this plant material. Our biggest idea now is to try to come up with a standardized extract that we can really work towards doing a defined clinical trial and getting some good information out there, and hopefully taking it through the botanical drug pathway that the FDA now has available to us to hopefully get out there and be a substitute for methadone or buprenorphine as a choice for physicians to use for opioid withdrawal treatment. And so that's the direction we're headed. Nigel, I'd be very interested to talk to you about how you got to where you got to, because we're sort of in that early phase. But we've done a lot. We've worked a lot on this. It's been a huge team effort. This is Ed Boyer, the physician that I've worked with since the beginning of this. He's standing next to one of our 2-year-old trees at Mississippi that we grew. But there's a huge host of people that I have to thank. And actually, the National Institutes of Health I have to thank a lot, because they actually helped fund this indirectly. They didn't really know it. They were funding it under our large center grant for natural products for neuroscience. But thank you for your attention, and I'll be happy to answer any questions. Dave. This one? Previous? In a 2-hour session, yeah. So, and that's very possible, because one of the latest papers reported it in vitro as being a partial agonist. But we haven't seen that in vivo, actually. And what's even more interesting, I don't show the reversion with blockade of selective antagonists. This is a mu-delta interaction, which is very interesting, because all the literature says that mitragyna doesn't interact, and 7-hydroxymitragyna doesn't really play any role through the delta receptor, but you can block almost the entire self-administration with a delta antagonist. Right, this was percent displacement, exactly. These were at, whoops, skipped it again. Anyway, it was at 1 micromolar. There it is. Right. Right. And so I respect that, and I also put that in all my grants now. This I did in about 2005, actually. So it was long before I knew about the PDSP. I know it was free then too. Yes. Actually, we have fingerprinted all of those. One other thing I forgot to mention, it's interesting in the United States, all of the materials that are in the United States are dried leaf materials. All the traditional use is through fresh leaf material. But we now have access to white vein, green vein, and red vein fresh leaf material, and we're having extracts made in the University of Science, Malaysia, to start looking at those directly. Yes.
So people are grinding up ligand and mixing it with Kratom?
It seems to be. I mean, I can't say for sure that that's what's happening.
Or unless the person was taking both.
Mm-hmm.
It doesn't make sense that you would add ligand to plants.
So the only thing that I can say to explain that is is that if you have a product on the market and you want your product to sell better than other products on the market, you make your product have a little bit of marketed advantage, give it a little extra kick that you know is going to work and cause repeat sales. It's interesting because yeah, if you try to track down any of these commercial products, you end up going down wormholes, and you never get. It's kind of circular. You never really get to find out where they came from, and it's very concerning. It's really concerning from the fact that you don't have any age restriction on it either. So anybody can go in. I have young kids. My youngest is 11. He can go into the gas station and buy this stuff and walk out with it. Yeah, and it scares me half to death. I think the natural product is perfectly fine. I really believe that. I believe it's really beneficial. I've received thousands of emails from individual users saying that it's changed their life. They've been able to get off prescription opiates. They have been able to return to work, return to a normal lifestyle. They have a energy, a good sense of well-being. They don't feel stigmatized by taking an opiate or having to go through those kind of issues. And so that was probably a big part of the DEA's pullback as well, because they received over 23,000 communications when they opened up for public comment in 30 days. So, yes. Mm. Mm. Oh, so I'm not entirely sure that these products are really what they say they are. I mean, they're saying green vein, red vein, white vein, but if you look into the literature, the green vein and the white vein are essentially the same. At least that's what they were telling us in the farms in Malaysia, and it just depends on when they're harvested. And then the red vein is different. A lot of people say the red vein is much more powerful. Now, if that's true, we don't really know. But if we look at just the red vein versus the white vein in this example, the white vein had much more of the 7-hydroxy in it, if that's really the way that the natural products were expressed, which we don't believe it is. So I really can't tell you. And the other most common question I get is, which supplier is the safe one to use? Can't help you there either.
There's at least one commercial Kratom product from Canada, which the 7-hydroxy Mitragyna has been specifically removed.
I don't. I believe it's called.
FX or something like that.
Yeah. This one is from Canada. That's here, and I'm not sure. It's a liquid formulation. I'm not sure if this was that specific one, but this one has 7-hydroxy in it still. So there are a tremendous amount of products. This was only a handful of what I just literally bought one day on the way home, and there's at least 2 times as many available at the same place. So. It's kind of scary. Another thing that's interesting, and it's in the literature, is that the 7-hydroxy mitragyna is converted in the liver back to mitragyna, which is a weird metabolism because you would think it would go the other way. But we've also seen it in animals where mitragyna is converted to 7-hydroxy mitragyna in the liver. And so we don't know which way is which at the moment. Because there's conflicting reports of what we've seen. So. Yeah. So So in the United States, these can be put in through the dietary supplement. So since they're a natural product and since they've had a historical use as a natural product, they're allowed to enter the marketplace. There is no regulation on them, which is a problem. And so this is a bigger issue, I think, in the United States, not just Kratom issue in the United States, but a much larger issue, because you can go into a pharmacy and you can buy ginseng extract, and it may not have any ginkgoloids in it at all. Okay, because there's no regulation by the Food and Drug Administration on dietary supplements in the United States. And so it's the one area that's been able to circumvent all of this. And this is, I think, the bigger question. And every time I've been interviewed by the media, I've tried to push that message out, that we need to have some more formal regulation of what's there and standardization of natural products. Mm-hmm. Yeah, and I don't really know the history back, maybe. It probably started to appear in the early 2000s. Yeah, it's pretty much right next to your 5-hour energy shots in the in the stores. Okay.
Thank you so much.
Thank you.
Sir, that was great.
Thank you. Thank you. Thank you.
Excellent. So another excellent example here of traditional medicine, ethnopharmacology coming together with excellent science to sort out these things. It looks like obviously kratom has great potential as a medicine, but inherently because of its nature, there are also problems with it as far as the regulation. So like all of these things, it's a two-edged sword, and what we have to do is develop frameworks where these things can be brought into therapeutic use, but without harming a lot of people. And that's partly where I think the experience from traditional medicine comes in, because these people, they know how to use it in the context of traditional medicine. So thank you, Dr. McCurdy. That was just extremely interesting.